Thank you so much, Johan, Chris, and Mareth, for the invitation to talk to you guys tonight. And thank you so much to Dr. Garma for the great introduction. Tonight, I'm just going to be sharing with you and taking on a bit of a whirlwind tour of what it means to be a palaeoecological researcher in South Africa, with a focus on my geographic region of interest, which is the Cape Floristic Region. And to start off, I'm just going to set the scene in terms of the modern environmental and botanical context. And we're really lucky to have within this study area that I'm looking at 3 globally recognized biodiversity hotspots. And the vegetation types that are encompassed within these different biomes shown here in the map. In red, the succulent karoo, which is the only arid biodiversity hotspot. The purple is the fynbos, which we all know is highly diverse and high levels of endemism. And then the Albany thicket or subtropical thicket biome, which is particularly understudied in terms of the modern context as well as the past vegetation communities. So it's really important, as we all know, I'm sure, from a botanical perspective in terms of the current distributions of vegetation within this region. The Southwestern Cape, or the tip of southern Africa, is also really important from a climatic perspective. And this is because it sits at the interface between 2 different major circulation systems within the region, which is the tropical easterlies, which result in our summer rainfall zone. So the weather we experience in places like Johannesburg. And then we also have, quite uniquely, we have the westerlies influencing the far southern and western parts of this region. And that produces what we call the winter rainfall zone. So the winter rainfall that we're receiving now in the Cape in terms of flooding, that is all linked to the dynamics within the westerlies, which is sort of high latitude temperate dynamics. So from a botanical perspective and from a climate perspective, it's really a very interesting, diverse, and complicated region to work in. And we can recognize that all of South African biomes are really important from a biodiversity perspective, and that we of course need to conserve and manage these correctly. But in terms of looking for long-term baselines for conservation management, we need to have an idea of what those baselines are beyond our recorded history and our knowledge that we have over the last decade or years. And from a climatic perspective, we need a full understanding of the natural variability of climate systems to enable climate models to predict present and future climate as accurately as possible. And we need this natural variability beyond what was measured, what we were able to measure in terms of things like rain gauges and temperature records. And so to summarize that, we need records that document long-term ecological and climatic changes. And this is where people like myself really come in, and where we can provide information to help inform those kind of questions and those type of models. And the work that I did that Dr. Garma very nicely summarized falls within the field of quaternary science. And quaternary is a geological term for the last 2.6 million years, which for most of you working in conservation, sort of in modern contexts, sounds like a really long time. But if you speak to a geologist, they think of that as the most recent past. It's the last blip within the Earth history. And quaternary science covering this period, and really what I look at is more like over the last 100,000 years and the last 10,000 years as opposed to million years. This work that we do is really by nature very interdisciplinary, and we draw on expertise and work within a collaborative team that cover many different fields, subfields within quaternary science and linked through science such as archaeology, climatology, and botany. And our overall aim is to provide a deep time perspective of environmental change. And we do this using something called proxy evidence, like a suite of proxy evidence, to reconstruct past environments. And then the next step from that is to infer ecological and climatic changes from the evidence that we are able to generate and document. So sort of the research approach on a broad scale in terms of my work within the Cape Floristic region is that we utilize and seek out in the landscapes natural archives that encompass and preserve this type of proxy evidence. And the two types that I'm going to focus on tonight, and I'm going to present some very summarized results from, are sediment cores, and this is extracted from wetlands, and a fairly unique archive that we also work on called hyrax middens, and that's predominantly found within the mountainous regions of the CFR. So the Cape Fold Belt Mountains are where we find hyrax middens, and more generally, the coastal low-lying regions are where we are able to find wetlands that preserve sediments below them that we can extract and use as an archive. The sediment cores are more, in inverted commas, more of a traditional paleoarchive that's used across the world. Similar to an ice core, which you might be a little bit more familiar with, sediment cores we extract from below any wetland, and in wetter parts of the world, these will be like large and deep lakes, which we can get really high resolution, really long-term records from. But down in South Africa, we kind of really have to work with what we have, and often these are ephemeral wetlands that are wetting and drying phases within that, which means that our records can be a bit discontinuous. And for this reason, for the fact that we have these semi-arid environments in here, and that wetlands are fairly hard to find in comparison to other regions in the world, we also use something called hyrax middens. A hyrax is a dusty, like as you can see here in the picture. And I can actually give you an entire 40-minute talk just on the hyrax midden research. So I'm going to really breeze through this right now, but the concept is the same. The midden in this case is a accumulation of predominantly Dussie urine, because the communities of Dussies live in communities that use one toilet, one latrine, and this archive of not fossilized but preserved urine and pellets are then a mine for different types of proxy evidence that we can then take back to the lab and analyze. And this is over the last decade or so, this has really changed our perspective on specifically paleoclimate data, and the insights that we've got from this has been unprecedented in terms of the high resolution and nuance and detail that we can gain from using hyrax middens as archives. Okay, so then talking about the proxies that you find within these archives, the one that I focus on, and the reason why I'm a palynologist is because I work with pollen. Palynology is another word for pollen, study of pollen. And I don't just work exclusively in looking at pollen. I also include other different types of proxy evidence, such as charcoal, which gives you some ideas, for instance, about fire regimes, which are really important within feinboss systems. And we also work with geochemists, sedimentologists, and looking at things like plant biomarkers to get all different types of lines of evidence, which we then can get from one type of archive and then get a much greater environmental picture in reconstruction from these different proxies. But tonight, I'm just going to give you some details about pollen. And the reason we can use pollen to reconstruct vegetations, I'm going to go through that now, is because pollen grains that we can identify have a direct link to the plant that produced it. So if we see, for instance, these three types of pollen, which are the sort of iconic feinboss types, Protea, Erica, and Restio, we can then assume that there was feinboss in the region if we manage to identify these different grains. And the reason that we are able to identify these and the fact that they're preserved within these archives is because they are very resistant to microbial decay. So the outer layers, what you're seeing in this video here, is the outer layers of two different types of pollen, and they have amazing features and detail that the palynologist can identify in terms of the different holes on the one, gives you an idea of what type of pollen it is, and then we are able to identify these across something went wrong.
Just go back into share
Yeah.
screen, Lynn, and then we can quickly fast forward.
Yeah.
Thanks.
Something was bound to happen.
There you are.
Yeah, I'm going to just skip. You got the picture in terms of that video, and I'm going to move right on just in case it crashes again. So this schematic here in this slide summarizes the sort of thought process behind how we use pollen to reconstruct different environmental variables, starting from the colorful pollen in the first circle. Flowering plants produce pollen, large quantities in some instances, and these are generally blown in terms of wind transport into the archive that we end up extracting, such as a wetland. So it comes into a lake or a flay, it then settles down to the bottom, and then we are able to come and do field work, and we extract that sequence of sediment from below a wetland, and within that are the preserved fossil pollen. We then, once we've done the field work, we take it, which I'm going to show you a video at the end of this talk on, to give you an example and a feeling and a flavor for that. We then go into the lab and we throw a bunch of chemicals at our samples to get rid of all the sediment matrix around the pollen, to just retain the pollen grains that are then we are able to identify looking at their surface features and different morphologies. From this, we get assemblages. So for each time slice, we look at we identify all the pollen we can within those samples, and we put that together. And we can then tell if we have a lot of the one type of pollen as opposed to the other, that, for instance, there was potentially forest growing in the area at that specific time slice, or potentially it looked more like the karoo at that time because we see charoid vegetation type, pollen vegetation types there. From that, we take the next sort of step or inference, and we can then say, for instance, that because there was a lot of forest, and because we know that forest types have bioclimatic envelopes that require lots of moisture, aseasonal rainfall, those type of features, we can then say that the climate was potentially quite, the environments were quite wet, so high in moisture availability. Whereas if we're seeing something like some sort of karoo vegetation, we can say that it was potentially a lot drier at that time. So these are the kind of sort of things we can infer from different pollen sequences, sort of on a general sense. Now I'm just going to be giving you some brief highlights of the research that I've done over the last couple of years in terms of within a context of the Cederberg, so a mountain region, looking at some high-rates midden research from there. After which I'm going to give you a coastal wetlands example from the wilderness region. So looking at the Cederberg, and I'm not going to go into what this graph is all about. All I want to highlight is that the red and blue lines here, which are the stable isotope results over a period of time that the midden compass, which is the last 20,000 years, show great variability, and we're seeing, which is inferred to be changes in moisture availability. So we're seeing lots of changes. So we have periods where there's much more reduced moisture availability compared to where there's probably more humidity. And this was quite an exciting finding back in that day. This was some of my master's work, and we published a couple of papers on that, and it was really cool to see this sort of level of detail and change within the isotope signal for this time period in the central Cederberg. And even to the point where, with a couple more middens and some more analysis in terms of the isotope results, we started using words like extreme hydroclimate responses within the region. But then in sort of stark contrast at the time, and again, you don't need to look at the details here, but just the point being is that these are pollen diagrams, and the giant shifts and changes that you see in the isotopes were not evident in the central Cederberg pollen record that I had generated for my master's. And when I saw this, I thought, well, this is really quite a weird setup where we have very muted changes in the vegetation, but great changes in what we've inferred in terms of the hydroclimate and the moisture availabilities. And the idea that we came up with there, and this sort of translates for other records within the region, much older ones, where there's even less change happening within these pollen records, is that potentially the the fynbos vegetation, the mountain fynbos vegetation within these type of regions are able to withstand, or are quite resilient to climate change. The climates are changing, and in some instances fairly dramatically, but the vegetation response is somewhat muted. And because it has such wide bioclimatic envelopes and are more linked to the geology and those type of factors, we're not seeing the same level of detail that we're seeing in the isotopes, which is like an interesting message in terms of looking at multi-proxy approach and looking at different proxies and understanding what they mean and how they relate to each other. Then moving down to the coast along the garden route to the wilderness region, where I worked on for that German postdoc of mine, where we looked at coring all of the lakes within the wilderness region. And this is a picture of Elandvlei. And after doing some seismic surveys, the team decided that really what we'd expect from below Elandvlei, below that lake, is a couple of meters of sediment, so 10 to the maximum. And in 2013, we went out there, we had a floating platform and a piston corer, and we went to the center of Elandvlei, and we cored and we cored and we cored, and we ended up with a sediment core of 30.5 meters, which is pretty amazing. Like, I've never worked on a record quite like that before. It took me a hell of a lot of time to count the pollen for that. I think at the end, I think it was a 2-year dedicated just to this one core to process all the pollen and to count all the pollen. And when we looked at the record and we dated the record, it was the last 10,000 years, which is known as the Holocene. So it was a relatively short amount of time, which means it was a really high resolution record that we managed to generate many different types of properties. But for my contribution, it was the pollen record. And again, this is very much a summarized slide here, where some of the sort of take-home messages from the work within the wilderness region, from Elandvlei, the 30.5 meter core, and also from another sediment core that I worked on just further inland on a very different time scale and this is from Van Kerwelsvlei, which covered the last 120,000 years. We see that there was this mosaic of fynbos and forest pollen within this region. So if you read some of the sort of historical archives where people thought that maybe the whole of the Knysna area was a lot more forested during the past, it seems that it was more mosaic type of vegetation was always in existence here. This is from the pollen record, we can infer this, and that in fact we've got a lot more forest in the more recent years than we've had in the past, and this is linked to changes in rainfall seasonality and other climate variables. And so what we could probably expect to see, which we know from other more modern botanical perspective as well, but this is reinforced here in the pollen, is that with warming and drying trends that may happen within the current and future, we will see less and less afrotemperate forests, and these will be more in refugia where they are able to withstand climate change, and more resilient fynbos types coming through instead of and replacing some of the forest within the region. Of course, there's also confounding factors within this region in terms of habitat transformation along the whole of the Southern Cape coastal lowlands and alien vegetation, which are complicating and causing vulnerability to and reductions in biodiversity that are linked to climate change as well. And then in terms of the research that I'm currently working on, so I'm not going to be providing you results of this as it's all in the pipeline at the moment, but just to give you some sort of an idea of what's happening right now in my lab at Nelson Mandela University, and the work that I'm currently doing together with my students is that we're working on a couple of sequences from the Baviaanskloof. So we're trying to address the lack of research in terms of palaeoecological work on the sort of eastern side of the CFR, and that's in the form of a Hyrax midden as well as a sediment sequence in the Baviaanskloof. We've also just come back about a month ago from the Swartberg, where we've identified many new Hyrax middens, and I have sediment cores that I've taken during fieldwork campaigns over the last couple of years have resulted in really cool sequences from Pearly Beach, from Plettenberg Bay, from a wetland called Whale Rock, and we've also got a 5,000-year record from taste print that a student of mine is working on, and I'm working on the pollen. And so those, we've got a couple of others as well, but those are the key sites that we're looking at right now, that in the next couple of years, hopefully we'll have papers out with multi-proxy analyses of both pollen, charcoal, and some other types of analyses that we're working on in our lab. Also, a new development is that I'm collaborating with some archaeologists, and this is happening from this year and into the future, and working on 2 key sites, sort of on the boundaries of the CFR, one within the Sakhalin Karoo, and one within the Cape, the inland Southern Cape, from near Kango Caves. And the idea here is that myself and my lab and our researchers, we provide the environmental context to the archaeological research that is done at key sites, to cave deposits. And this is really important to link the archaeology with environments and climate change, to see whether the innovation and the technological changes that are found within these sites during specifically the Middle Stone Age are potentially linked to changes in climate or environments within the region. So we are working on those in the next couple of years. I'm also trying to tie together more closely paleoecology and ecology, and I'm doing that in 2 specific ways at the moment. I'm working on, together with the PI from Sanby, from a project that's looking at Asteraceae pollen, because we have a high amount of Asteraceae, which is the daisy family pollen within all Southern African sequences, pollen assemblages, but we normally only identify that as Asteraceae, which is a giant family with cosmopolitan types within that. So if we are able to just discriminate the different types of Asteraceae to a genus or even potentially a species level, we can then really manage to mine old sequences and new sequences for much higher ecological detail that we can gain from that. We can tell the difference between different genera of Asteraceae that like different types of climates and different environments. Then we can then make those inferences from that. But we can only do that after a fairly extensive project that's currently happening, and which requires, this is my flagship microscope in the Paleo Lab. It's is an inverted Nikon light microscope with fluorescence and automated stages and micromanipulation capability. So we can isolate individual grains, which this is a student of mine, Luke, who's working on this project as his honors project. He has to sit here and measure tiny, tiny little bits of these pollen grains so that we can then amass a data set and look at it statistically to work out whether we can tell these different Asteraceae types from each other in a robust manner. So that is currently in the pipeline. I'm also looking at when we have set up pollen traps, which stay in the landscapes, in this case in Pearly Beach area and within the Nelson Mandela Reserve, where we are looking at a more sort of modern pollen calibration work, where we're trying to see if different vegetation types have different pollen signals. And so this is sort of on a sort of short temporal scale, but we're linking it to our records within the area that are over long periods of time, so that we can tweak different taphonomic responses of the pollen types. For instance, what are overproducing, what are underproducing pollens, so we can kind of calibrate the assemblages that we generate on the sort of fossil side. And that work has been done by a master's student of mine. And then I just wanted to highlight that it's really important to archive, and for sort of a selfish perspective from a paleontologist, to have your records robustly entered into a database that's going to be safe in the future, and also from a community perspective, so that these can be used for larger syntheses and integrated into larger studies to look at vegetation responses across the whole of South Africa. I archive all my data in the African Pollen Database, which I'm a data steward for, and that feeds into a larger international database for paleoecology work, which is called Neotoma. So I'm working to develop and make sure that we archive not just mine, but all South African palynology records within this database that's then going to be freely available and open source and online. And this slide is just to give you an idea about the great scope, both current and future, for the Paleo Lab at Nelson Mandela University. We are not just working on pollen, as I mentioned before. We're looking at a suite of different proxy evidence, which is the blue sphere side of branch of this mind map. Many different types of proxies which are really useful, such as diatoms, looking at diatoms and fossil records from the coastal wetlands that we have, as well as I mentioned before, charcoal in various forms to look at the archaeobotanical work done and more in archaeological deposits, and then the micro and macro charcoal, which are found in sedimentary sequences. And we also, which leads into what I mentioned before in terms of tying modern pollen vegetation calibrations, looking at the ecology and linking that to the palaeoecology. That's the turquoise sphere. And then the last kind of different application for pollen that I want to mention today is the modern pollen monitoring. And that is work that we're doing in a South Africa-wide network called SAPNet, South African Pollen Network. And the website there is pollencount.co.za. And this application is for allergy sufferers. So we work within the medical industry here, and we produce PE's only pollen report for each week we put out on this website, and it's for many of the cities in South Africa. When different types of allergenic pollen are high, you can tell on the dashboard that's on the website, and then you know you need to take your allergy medication, or you shouldn't go for a run when the grass is really high. These type of things. We provide a public service here, and that's work done in collaboration and led by the UCT Lung Institute. So yes, every week, check out the pollen count for any of the major cities in South Africa to give you a real indication of allergenic pollen seasons. If you look at your weather app on your phone, you will see a thing saying pollen index or something like that. That is completely made up. None of that data, there's no data behind the inferences put out there. So you have to come to our site to get actual data that we are measuring on a weekly basis. And that's the picture here is of the spore trap that we have up on the Ocean Sciences campus at Nelson Mandela University. And also just to mention that all of this work is very much predicated and linked to student training and mentoring. And I'm very grateful for the great student cohort that I'm currently working with. And we have a lot of fun in the field, which I'm about to show you now in terms of of how we go about doing field work and what kind of equipment we use for that. So this is a video compiled for a sediment coring exercise that we did for an internship project last year in the Zeeberg in the Eastern Cape. And we worked with the first sequence that I'm going to show you now is using something called a Russian corer. So this is us out in the field, and that's us extracting the sediment using the Russian corer. And then the next piece of equipment we use here is called a vibrocorer, and that gets you much longer sequences, and it's a little bit more involved to set up and extract. And it uses a Poco vibrator to put onto this on the tube there, as you see here. So this is the vibrating part, and then you have to hang on it and push it down into the sediment, and then you use the tripod that we set up here to extract the cores from there, and then wrap them up. And make sure we label everything correctly. And then take it back to the lab. And that's my whirlwind tour of palaeoecology in the Cape Floristic region complete. Thank you very much.
I see a lot of hands going up there. Let's go with Terry first. Terry, please unmute. Thank you very much.
Lynn, thank you. That you're doing some amazing work and a lot of different aspects to paleoecology, which are really, really necessary to understand the pollen itself. There's a lot of other factors around it, and you're doing all of the work around it to really understand what's going on. It's really impressive. It takes me back to my former life as a paleoecologist.
Oh, wow.
But I'm curious about the hyrax middens. Two questions. One is, what is the proxy that you're getting from that? Are you using chemicals in the urine, or is pollen actually falling in there and you're extracting pollen? And then the second question is, are they really continuous for very, very long periods of time, or do you have gaps where maybe the hyrax move out of the cave for a while and then they come back in? Or are they generally, do they stay there for generations and generations and have a fairly continuous record?
So to answer the first question, yes, there is pollen. So I do the pollen within that. My collaborator and mentor, Dr. Brian Chase, he spearheads the, so you know him.
Yeah.
So he does the stable, his forte within the hyrax midden work, and I mean, he is the hyrax midden guy. He does stable carbon and nitrogen isotopes. So that's the key hydroclimate proxy that we look at. But there is pollen, and beautifully preserved pollen. It's much nicer than working on the sediment cores from the coast, which is very discontinuous preservation in terms of the pollen, and discontinuous in terms of the chronology and the sequences we get because of the sort of wet, dry nature of those types of sediments. The hyrax middens themselves are super, I can't say all of them are, but everything we've worked on is super continuous in terms of the accumulation of the actual material. The hyraxes use a rock shelter until they fill up. So they literally are using it until it fills up. The community hangs out in one place and uses it. And then once they can't fit literally into the space, they will leave. So we just have to date it quite well. And then you get extremely high resolution. I mean, I'll just share with you, just out of interest, that I flew through this one. But the actual laminations within, within the hyrax middens are pretty phenomenal in terms of the fine resolution of it. Sorry, hang on, it's going to crash again. Oh my gosh. Bear with me.
So how long is that hyrax midden record usually?
We have records beyond the ability to date it with radiocarbon. So we have over 50,000 year records and basically continuous for 50,000 years.
Wow.
So it's a game changer in terms of Southern African paleoclimate work, really. I'm sorry, now I'm like lost myself in my own computer. There we go. Let me just get to this. Sorry. So if you see here this image, this close up, this is using a microscope of a piece of hyrax midden. This is 2 centimeters. So we're talking about less than a millimeter of like laminations, and each lamination, if we could date it to that degree, could give you a time slice at that resolution. And this is from Namibia, so they look different. The Cape ones are blacker because of the vegetation. So this is the Namibian ones. You can actually really see the lamination. So it's like a speleothem, basically.
And are the laminations generally 1 year, or?
I mean, no. So they're not annual. You'd need to date them to figure out that. So it's not a VAV sediment, but it's almost a VAV sediment. You could date it to that degree. Yeah, so it's great.
Yeah, thank you.
Pleasure.
Terrific talk.
Thanks, Terry. I think.
Sorry, Chris.
No, I just want to say thanks for an interesting question and interesting answer. Shall we go over to our friend Marty Jasper?
Go for it, Marty.
Marty, the floor is yours.
Thanks for a very interesting talk. I actually attended a talk on pollens at the at WITS at the Origin Center, and it was very interesting as well. I just wanted to, so you're mainly talking about in sediments in wetlands. I was wondering, what about in other archaeological digs, like, for example, at Mapungubwe, with those ancient civilizations and that sort of thing? Do you also get pollens out of there that you can do analysis on? Or is it only basically because of the type of vegetation that there aren't the same sort of vegetation, so you aren't able to get the same sort of pollens? Or yeah, I'm just interested if this can work on all archaeological digs and you can go afterwards and find stuff that had never been looked for before.
I mean, in theory, it depends on the preservation. So there's certainly, if there's flowering plants in the landscape that produce pollen and are dispersed in a manner such as wind and will be included within any type of archive, if it's an archaeological deposit or, yeah, like a cave sediment type of thing, you can potentially find pollen and work on it. Unfortunately, they're not as well preserved in terms of the environmental context, and we're often unable to get enough pollen that's preserved within those type of sequences to work on it. But in that case, we use different types of proxy evidence, such as phytoliths, which will still give you an idea about the vegetation, just in a different form. So the answer is yes. We don't just work on those type of archives, but those are the most prevalent in the regions that we're in, and the most successful in terms of extracting pollen.
And then the other sort of question that I had, I mean, there's whole areas, for example, of the coast, on the east coast, where it actually all used to be underwater, and is now, dry land in parts of Mozambique, even up to Kruger and that sort of thing. Is there similar sort of technology to try and date those things, or is it also linked to pollens? I mean, how accurate? What is the level of accuracy of the dating? Because I mean, sometimes you hear, but up to 10,000 years or plus minus, which seems quite crazy. Yeah.
It depends on how much money you have, to some degree, in terms of how many dates you can process and pay for, in terms of radiocarbon. So there are definitely ways to date things to a higher degree. It's not tens of thousands. For my sort of error bars, like maybe 1,000 maximum as an error on a date. For instance, something that's 15,000 years, you might have a 500-year error. We work also in marine environments, not myself personally, but paleontologists also work on sediment cores that are extracted below the ocean. And that's also very much doable. It's just about having research vessels go out and extract them, and again, looking at the preservation of the pollen to actually get a statistical amount in order to make these inferences about the vegetation. Which is also, and we also, in terms of just going back to the dating, it's not just radiocarbon that we can use. If we're looking at sort of high resolution, shorter time periods, there's something called lead-210 dating, and cesium and lead dating, which gives you an accuracy of much finer detail on the temporal scale. So it's all about like knowing what questions you want to answer and what the archive itself is encompassing. Sometimes you have to start a bit blindly. You go out and you take a sediment core. You often have no idea how old it is, and the sort of protocol then is to take a basal age and kind of work out what you have from that, and then a sampling strategy that encompasses the right amount of time for your samples to give you the right assemblage data in terms of the pollen and to answer the questions you're looking at.
And is all the dating basically done locally? Is the machinery here, or is it sent overseas for dating stuff?
Up until recently, it's been a safer bet to send it overseas. But at WITS, at the iTemba lab with Stefan Woodwarn, he's doing a great job on sort of resurrecting the ability for South Africans to go to a South African institute and get radiocarbon dating done. I've in the past often sent my the samples to a lab in Belfast, in Ireland. But yeah, it's a very pricey affair. So often when you're looking at research grants, the biggest contribution to the budget is radiocarbon dates.
No, that's why I was asking. Hopefully it can get done locally. And I mean, you don't hear of people studying to do that. Is it like a whole degree on its own, or how do you get into that? It's quite interesting.
And yeah, I wouldn't know how you directly get into that, but again, Stefan's doing a great job at ITEMBA at WITS. You and I need to actually do that this year. You can send a student over, and he will. And we've done this last year with my head lab tech. She went over and learned the process from him on his equipment over there. And he doesn't charge for radiocarbon dates if you are working in collaboration with a student and you send the student over to run the samples themselves, which is a really great initiative.
Oh, that's awesome. Thanks very much for answering my questions.
Pleasure.
Thanks, Marty. Always interesting to listen to your questions. Appreciate it. It adds value to the broader discussion. Thanks, Marty. Pippa Haroff. Pippa, I'm going to unmute you. Pippa is from the West Coast Fossil Park, so she also somebody who's done some great work there. Pippa, so if you just accept unmute, and then we get you. There we go. Thanks, Pippa.
All right. Yeah, thanks very much, Chris, and wonderful talk. Thank you, Lynn. I've got 3 different questions. Back to the dassies. I didn't quite get why the urine preserves. What's the preservative element in it, if you know what I mean?
So it doesn't have it preserves because of the environment. So dassies wee everywhere where they live, and it often isn't preserved because it gets washes. It's too wet, or there's no sort of like environment for it, or home for it to stay and get preserved. So it's mainly the environmental context of the latrine. So the fact that they're in these rock shelters in places like the Cederberg or the Baviaanskloof, that they are protected from being washed away or decayed. So they don't really, I mean, they're quite well preserved in general, but it's a matter of its context that that we are able to then go and extract it as a set thing.
Okay, so there's no...
I mean, maybe I actually don't know if there's some specific chemical in there that is particularly resistant to decay or not. I think it's just the fact that it's able to sort of hide away from environmental degradation in areas like rock shelters and caves.
Okay, thank you. And then the amaryllids, do they show up well in the pollen record?
Amaryllidaceae, yeah. The one sort of caveat on in terms of pollen analysis is that we can't often discriminate as to the species level that a botanist can. So we can often tell pollen only to the genus level, family level, because the pollen grains look so similar. And it just takes too much time and possibly to not get to something as specific as a species level.
Yeah, okay.
But they are very nice. They are nice pollen grains in terms of morphology, quite easy to, all the sort of like monocolpate ones are quite easy, like lilies and amaryllidaceae, and those guys do definitely show up in the records.
Okay, thanks. And what's the oldest pollen to date in the world?
I mean, it's as old as the oldest flowering plant.
Okay.
Millions of years.
Yeah, like 60 million odd. Right.
Something like that, yeah.
Actually more than that, yeah. Cretaceous.
Again, it's about like if you have the environmental context, if you have something preserved that you can find. So we have Miocene. I'm not working on it, but Frank Newman is working on Miocene pollen in the Cape, which is obviously on a whole different level to the quaternary work we're doing.
Okay, thank you.
Pleasure.
Thanks, Pippa. Thanks, Lynn. Fascinating. Putting you on mute, Pippa. Dr. Llewellyn Taylor, asking to unmute. And Marty, I see you've got another question. You're welcome. After Llewellyn.
Yeah, sorry, Lynn. On average, what depth do you normally core to in wetlands?
I mean, it very much varies. From the sort of average is about 4 meters, if that, maybe like a meter, totally depends on the wetland itself. But that's why the 30.5 meter core that I worked on was pretty phenomenal, because before that I'd only worked on things of 4 meters was long, whereas a meter, often sometimes less than a meter, you still get very valuable information on those type of shorter sequences, and you can ask different questions from sort of like more recent paleoecological work. Whereas things that are like the 4 meter core that I got from Van Kervel's Vlei in the wilderness area is pretty fascinating, because it covered such a long time period to the last interglacial, which is 125,000 years ago.
Yeah. The reason why I ask you the question is I normally call for wetland delineation. And when we do wetland delineation, we normally go to about 0.5 meter. But clearly all we're looking for is manganese and iron oxide mottles and so forth. And yeah, very often when I'm doing it, I think to myself, well, damn, should I be going deeper? But, you know, there's always a limit to time, a limit to what depth you can get to with a handheld auger.
Oh, okay. All right. Yeah, no, that, I mean, it's fairly tricky and takes quite a lot of effort often to little reward to go much beyond that in these sort of South African wetlands that we have, unless you're looking at a giant deep lake in tropical Africa where you're getting literally hundreds of meters. It's a whole different ball game up there.
Oh, great. Thank you very much.
Thank you, Llewellyn. Marty? And then you're on after that. I just want you to take over in terms of next week's speaker. Putting you on mute. Yeah, you're mute, Marty.
Cool, I'm unmuted now, thanks. Then just in terms of some of the pollens that you have found, where you aren't able to identify them, is that kind of a chance that it's actually of the species that's almost gone extinct or no longer around? And then the second part of the question is the fact that, I mean, pollens are part of the whole reproductive process of plants. Could you almost bring back to life some species that have gone extinct, or are they kind of neutralized now? They're no longer viable reproductive seeds, or whatever the right word is. But yeah, have you found the extinct species from what you've found, or you just kind of say, well, we can't identify it now, but maybe in future we can?
You never really look at things. Everything that I've worked on is existing flowering plants that we can, if I can't identify to a very high level, I pretty much know what it is, or someone within the community will know what it is. I've never heard of the case that we've come across completely foreign pollen grains that have a morphology or a connection that we can't like tie down to something that is existent. The second question, what was that about?
Well, so if that's the case, then it doesn't really apply, because there's nothing that you find that's extinct that you could bring back. So
yeah, no, we don't. You can't do sort of resurrection ecology on fossil pollen, because it's the only thing that's preserved of the pollen grain is the outer layer. So none of the cellulose or any of the reproductive material that could then propagate the species is still around when we look at fossil pollen. It's basically a skeleton of a pollen grain that we work on. And also within the processing steps within our lab, to identify that outer layer of the grain, we get rid of anything that is there so that we can just see and isolate the outer layer and the surface texture of it to be able to identify it. So yeah, you couldn't do much in terms of that type of stuff on a fossil pollen grain.
But it is interesting. I mean, if you talking about so many thousands of years, 60,000 years or whatever, for that core that you did 30 meters deep or whatever, but there's nothing found there that's not currently present today, which, I mean, I don't know, there's supposed to have been lots and lots of things going extinct over the years through different weather experiences and climate and whatever. So it is surprising for me.
It's also the resolution that we identify things are not to the species level. So potentially things that could potentially have gone extinct over the timeframes we're looking at, which is fairly rare in itself. I wouldn't be able to tell you to that degree because I'm only looking at a family or a genus level.
Makes sense.
Cool, thanks.
Thanks, Marty. Thanks, Lynn. Johan, over to you.
Lynn, may I ask, you mentioned
Yeah.
dust were 50,000 years old?
So that's the sort of the limits to radiocarbon dating. So dusty middens, I'm sure that they are definitely older sequences, but to date, pun intended, we only have records to about 50,000, not only because that's pretty phenomenal, but that's the kind of the range we're looking at the moment.
So may I ask, did dussies look more or less the same as today?
The dussies have been the same, yep.
Okay.
we can, and the middens are the same within the area, depending on whatever vegetation they're eating.
Okay.
Fantastic. Tim Hoffman, please come in. Just unmute yourself. There you go. Hello, Tim.
Yeah. Hello. Hi, Lynn. Nice to see you again. And
Hi,
thanks
Tim.
so much
Lovely
for the
to
talk.
see you.
Thanks very much. Very interesting. Beautiful graphics. Lovely to see everything. I want to ask a few questions about the ecology. In the sense that Frank Newman did some work on the Princess Flay, and he looked at charcoal and made the case for seeing a signal of maybe not hunter-gatherer, but certainly early pastoralists 2,000 years ago, and then a much greater incidence of charcoal coincident with the arrival of European settlers in the Cape. And I wonder if you could just say a little bit more, because fire is such an important dynamic and determinant of the proportion of forest and fynbos. We all know about the big fire in the 19th century, and even the big fire recently in Knysna has a lot to do with aliens, but fire is a very critical driver of forest fynbos dynamics. And I wonder if you could just say a bit in your work on that long core and other work that you've done, and whether dusty middens also can pick up charcoal, and whether you're looking at that in dusty middens. So just something about fire and the fire dynamics in your work would be really interesting.
Absolutely. Yes, so I mean, it's a very good point, and I didn't touch on it in this talk. All the sequences that I worked on in terms of generating the pollen, we also look at something called micro charcoal, which is a sort of like very much a sort of regional, we can compare that to sort of regional fires. Recently within my new lab, I've started also looking at the sedimentary macro charcoal for all of these sequences. And that gives you an idea of more local, so at the point source fire. We can reconstruct the fire history for that specific site. And then compare it to the micro charcoal for a regional perspective. The key here is to try and get high resolution, and that's what we're working on. And then use something called char analysis, which is a peak analysis where you can pick out, because there's always going to be background charcoal in sequences, and it's like trying to pick up where there is like significant fires. That's quite important. And we are working on that, and some of my students are generating new macro and micro charcoal sequences to go hand in hand with the pollen, so that we can look at these type of ecological questions and pull out, are we seeing, because sometimes it's not, my sort of initial inference is always like this, the fynbos sum is going to match the charcoal sequence, and we're going to see higher fires at the same time as more fynbos. And sometimes it's not quite the case, and I think with the elements of climate within that, it's quite difficult to disentangle some of these signals. But it's certainly an element that we are working on continuously. In terms of the sort of the anthropogenic signals within these sequences and the links to fire, we're also working with coprophilous fungal spores. So looking at the spores to pick up pastoralism in the landscape on longer time scales, like large herbivores. And that's a really cool new sort of line of evidence. And then if we had, which is something, the work that my student who's doing a master's and presenting her proposal next Monday will be doing, is tying in the charcoal with the fungal spores and the pollen, at taste print over 5,000 years to see kind of these dynamics between them all. But I think a lot more work can be done, and not just generating the micro charcoal at the same time as the pollen, but very much looking at similar things to what Frank put out in his Princess paper.
Can you get charcoal and spores from middens as well?
Yep, that's also what I'm working on right now, because at the time that I looked at my master's midden stuff that I didn't think about the fungal spores. I've always done the charcoal, so you do get charcoal within the sequences. It would be interesting to see kind of how taphonomically it plays out, like how much charcoal is coming into those type of midden sequences opposed to a much open setup for a wetland. But we have the data for that, and we are now generating the fungal stuff also for the middens, which will be another interesting sort of look, because obviously it's a midden that is sort of like got the coprophilous double layer then in terms of it being coprophilous in itself, and then also encompassing spores from outside in. So I think there's going to be some calibration studies that need to be done on that. But yeah.
Thank you, Lynn.
Pleasure.
Yeah, sorry, Lynn, if I could ask now again. I just want to find out from you the pollen database that you showed us on that one slide. Given that I'm from the eastern part of the country in the Lowveld, there seems to have been no data for the area. Am I correct in assuming that, or why? It's just I'm just curious because I saw that immediately.
A good observation. I mean, there's very few. Do you know of big wetlands in the area? And if so, yes. I'm coming to them.
Yeah, well, most definitely. If I do a lot of wetland specialist work, so maybe we could chat. Yeah, most definitely. I noticed it immediately.
It's eastern, on the eastern side. There's sort of concentrations around where the universities and where the research is happening. Access, like ability to go and explore areas, known areas, unknown areas. So if there are wetlands out there, I don't think it's a case that there's no pollen. It's just that we haven't explored some of those regions, which we really should. Sure.
Okay, well, let's definitely speak about that, because we can certainly assist you in that. Yeah, and then just one comment. I've just read a book called Kindred, which is an incredibly detailed book on Neanderthal man, incredibly detailed. And what amazed me about the kind of work that gets done on the caves in which the Neanderthal man lived, and the details and the absolute resolution and the incredible work that is done to look at those levels. It would be great if I just don't know how much the kind of work that you do would have actually been done in caves like that. But this is mostly in Europe, obviously not in Southern Africa.
It probably would be the case, but again, cave deposits are kind of notoriously not great in terms of finding pollen. So specifically pollen is kind of hard to come by in these things, but I'm sure they would have tried in these really high profile sites. Yeah. But that's another thing that I haven't even included it in the sort of mind map of what we're doing within the Paleo Lab. I'm also trying to start up phytolith analysis and train students to identify that, because that is found more often and can give you an idea about the environment. But again, all these archaeological sites, a large component of it is to reconstruct the environments, various ways of doing that. I have a postdoc that's looking at the sort of macro level charcoal, so that you can identify to species level, and bits of like identifiable charcoal by the naked eye in archaeological deposits. There's a whole different science to how to identify that, and that gives you species level data, and that's often done in archaeological contexts.
Liesel van As, I think you're probably going to be very much close to our last person. We're down to 28 people. Golden rule is on 25, we kill it. So they're still going to hang in there. Liesel, over to you, please.
Okay, first of all, I I never do this family thing, and the dog is now sleeping. So, just to Marty, and I'm sure Lynn will agree with me. It's like bringing back the mammoth. Habitat has changed, and whatever we have now, they will not survive, because we also don't know what other symbionts they had in the stomachs that they need to survive. So even bringing back any vegetation, that can be a tricky thing. And then I want to ask Lynn, Bruce Rubich and Terence McCartney wrote a book, Earth and Life, and in it they refer to the super swell, meaning the tip of Africa is actually lifting up. And if you look at the graphs that you have with some of the places where you sampled, obviously the majority were inland, but there were a couple of them literally on the edge of the continent of our, the continental shelf. So my question is, what is the influence of the lifting of the African tip on the pollen?
I don't think there is any influence on that. I mean, that's on a sort of geological time scale, and on the things that we're looking at. I mean, I'm no expert on that type of level of geology, but I would not think that it would have much of an influence.
Okay.
Thanks, Liesel. All right, anybody else? Richard Masabedi. I'm gonna, yeah, you can just unmute yourself, Richard. Sorry. Just unmute, Richard. Yeah,
hello. Hello. Thanks, Lynn, for a very insightful talk. I don't have a question. I just want to make an impression. So I work in Twihaba caves. So I heard you, I think you kind of demonstrated the potential of caves in reconstructing past climates and environments. What do you think, is there potential in using bed guano piles, do you think?
Possibly. It's a good question. I don't know how, if they sort of accumulate in one place long enough type of thing, or if it's like ephemeral and you just get sort of little snapshot. But I mean, sort of to give you a backstory on the Hyrax midden work, it was inspired by work done on pack rat middens, which are more like nests and more like time capsules from one period of time, which are not sort of fine layered accumulations like the middens. So you could potentially look at guano in that way if it was in a cave, and it certainly would be the nature of it would be good enough to incorporate pollen, depending on the sort of opening of the cave and that type of stuff. Like the idea of you'd get pollen in there if it was dispersed there. So it depends on kind of the setting of it. But I quite frankly have no idea. I've never really thought about that.
Yeah.
Interesting thought.
No, thank you very much. The other thing I want to mention to you is that I also work in the Okavango Delta. And the challenge with coring the Okavango Delta, getting cores, is that it's overlaid on the Kalahari sands. So the deposits don't stratify properly because of the sand. Yeah, I just wanted to make an impression. I do stable isotope research. And recently I got some support to study the food web of Twihaba. And I haven't analyzed stable isotopes of carbon and nitrogen in Southern Africa. Do you have a suggestion as which probably the cheaper lab to go to?
So we run our carbon and nitrogen isotopes often through UCT, through the archaeological department there. They have a stable isotope lab. I haven't done for a while, and I don't know how expensive it is. It is a problem it's time consuming and therefore expensive to process. I know I've weighed out tiny little things for isotope before. UCT isotope lab. Often the way to do it is to try and strike up a collaboration with somebody that has the lab, and then as long as you include them on the papers and you keep them in the team, they could potentially do it for free. And that's also worked quite well in the past. But yeah, I don't think we run that at Nelson Mandela University, and I don't work specifically on the isotopes myself. So yeah, this is just my thoughts.
Yeah, no, thank you very much.
But I think the person to speak to maybe is Dr. Vincent Hare, who's working and potentially running the isotope lab at UCT.
Dr. Vincent Hare?
Hare, yeah.
Okay,
I can chuck that in the chat.
Ladies and gentlemen, we're down to 24 people, and we thank you so much for your participation, and thank Lynne for a very insightful and a lovely discussion and encouraging discussion, because, you know, if you go that deep, you're going to learn a lot.